Hi guys, and welcome to Learn Extra Live. This is Life Science, and it's Indy and Kathy with you today. Grade 11s. Today is an open session.
So revision.
Revision on everything you guys have done. But what this means is that they can send through questions on anything that they've been struggling with, what they've done so far in school. So guys, please make sure that you do that. Get linked with us. Chat to us on Facebook, facebook.com/Learn Extra. Also chat to us on Twitter @Learn Extra, and let us know if there's anything that you are struggling with and you need us to help you out with. I think, let's have a look, Kathy, and see if anything has come through from the grade 11s. Okay, hold on. And I know Sedimo has been posting on the page, and she's waiting for the grade 12s, and we are going to be going through evolution, guys. But also, grade 12s is also an open session, so start sending those questions in now.
Well, I think, you know what, people, we've got 2 sessions left that have been scheduled before you write or before you go on holiday. So I'm quite happy to change whatever. I'm going to start with evolution and the theories, which we haven't done. I'll go through that, and if anybody has anything urgent they want me to go through, like the excretory system or whatever, I mean not excrete, that's grade 11, DNA or genetics or anything, just put it on and we'll stop and we'll go through that as well. Okay,
well, Leanne says, can you do cell division? Is that part of grade 11?
Grade 11? It was mitosis.
Yeah, but we need to know, guys, what we need to know is we need specific questions. So
no, not specific, but I mean you can, division. Cell division would be mitosis. So just give us an indication if that's right, and then I'll do mitosis. I'm cool with that. Okay, people, so we've been asked to do mitosis. Remember, in grade 12, you're going to do meiosis, which is sexual reproduction. So just to help you here, you've got cell division. Okay, there are 2 types of cell division. Okay, the first type is mitosis, and the second type is meiosis. All right, now, the way to remember it is toes grow, whereas mitosis is meiosis, which is sexual rep. So it's for sexual reproduction. Okay, so if we go through mitosis and my toes grow and get too small for me, we do it like this. When does mitosis happen? Okay, why? Mitosis is for growth, for repair, for replacement, and for reproduction in unicellular organisms. Okay, so growth, we grow bigger, your hair carries on growing, your nails grow, we grow. But at some point in human life, or any organism's life, you get to a point, animals by the way, you get to a point where you've now grown a maximum amount. Okay, you are now an adult. So there's not a lot of growth that occurs after that. In our cases, our hair and our nails really, but we have to repair a whole lot of cells. So we damage cells all the time. And you know, they'll tell you that we've got all these cancerous things that all around that destroy cells and blah, 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 and pollution. We have to repair those cells. If you go to gym and guys are trying to build muscles and get big, all right, for sport, etc., what happens? The muscles first get broken, and when those muscles get broken and ripped and hurt and damaged, the body repairs them and it adds a couple more, and they will have a bit of growth taking place there. Okay, so you're repairing the old ones and you're building new ones to make sure that there are enough cells the next time around. And then the guys go back to gym after 48 hours and they break those muscles and then they build new ones. So that's for growth and to repair damaged cells and replace those that are so damaged you can't be repaired. They have to clear it out and then make new ones. So replacement and reproduction in unicellular organisms. Think, unicellular means one cell. So if it's only got one cell, well, hello, it hasn't got like mega reproductive organs. The chances are that that one cell meets up with another one cell is going to be very minimal. So what does it do? It makes sure, or nature has made sure, that that one single-celled organism is able to reproduce itself and ensure the survival of the species, which also goes a bit to evolution, all right, and diversity. So unicellular organisms are, you've got this one cell, and this one cell is now going to make two of itself. Okay, so with mitosis, the whole process takes place in 5 phases, all right? Although the first phase is not really a phase as such, it's just a preparation phase. So we start off with 1, which is interphase. And then we have prophase. And then we have metaphase. Anaphase, and the last one is telophase. Americans write it with an E, we write it with an O, it doesn't matter, either way, T-E-L-E, T-E-L-O, both are acceptable. All right, now, within these 5 phases, what is the outcome that we want for mitosis? We want to have one cell, and that cell is diploid. In other words, it's got a full chromosome count, and we want to make 2 cells, and those cells must be identical. We call them identical daughter cells. Now, can you imagine if I lose a cell in my eye, okay, in my eyeball, and one of the retina cells, maybe a cone cell, gets damaged, and that's it. So now I have a choice. I can either leave it a hole there, which means eventually I'll be completely blind, which is not the way the body works, or the cells next to it will now make a new cell. And when it does that, it must make sure that the cell is identical to itself, so that I don't end up with a heart cell pounding away in the retina of my eye. Or if I get a spot on my cheek and I go, like all human beings do, and I squeeze that spot and I fiddle with it, it's got to heal. I do not want liver cells developing or kidney cells developing here on my cheek. What cells do I want? I want cheek cells developing on my cheek. And people, it's winter. All of us at some stage have very, very dry, cracked lips. Can you imagine if a kidney cell had to now develop here, or a cuboidal cell had to develop here on your lip? It would look awful. It would look like you've got something growing on here, or a heart cell. You'd have this pulsing on your lip. We don't want that. We want the same cells to replace the ones that have been damaged. So we want a diploid cell, and we want identical, and we call them daughter cells. Identical daughter cells to develop. So they must be exactly the same as the original cell. And that is why we use mitosis for growth, for repair, for replacement, and for reproduction in unicellular organisms. So here are our 5 phases. Interphase is just a prep phase. Okay, and this is when all the chemicals that are needed are taken to the cell. So if we look at that cell it looks like any other cell. We can't say, interphase is happening. Something else, you must remember that if you're going to have a birthday party or have any kind of party, it takes weeks and weeks of preparation. The Queen's having a jubilee now. Let me tell you something, they've been preparing for this for the last year. They've been preparing for the parties and the celebrations. So if you think about your birthday party, maybe you plan 2-3 weeks in advance. And for 2 or 3 weeks, you and your friends are talking about your birthday party and talking about all the things you're going to do and what you're going to have and whether you're going, what you're going to eat and what food you're going to have, etc., etc. And you plan this party, where, what, how, what you're going to wear, etc. And on the morning you go and you get your hair cut and sorted, and if you're a girl, whatever you have done to your hair, and you get your nails done and you put makeup on and you get ready and you go buy an outfit. All of this is still preparation for a party. And then the evening is there and all your friends arrive and you have this wham bam incredible party. The party is over in 3 or 4 hours, but it's taken 3 weeks to plan for this party. The cells do exactly the same. This interphase is the longest phase. Okay? It takes up about 90% of the time that it takes for mitosis to take place. Why? It's the longest phase. Why? It is the preparing phase. So it's getting all the chemicals ready, it's getting everything sorted in that cell so that when the nucleus now starts to divide, everything is there and everything's in place to have the party. So the party is actually prophase, metaphase, anaphase, and telophase. Now it also sort of happens in our lives because you've got the prophase when everyone arrives. You've got the metaphase when everyone starts to mingle. You've got the anaphase when there's some people that are upset because things are not going the way they want to. And then you have the telophase. Everyone afterwards now sits and makes telephone calls and to let everyone know what happened. Okay, like telecommunications via cell phones and messaging. Right, so we have the actual party here. Interphase, just the prep phase. You can see nothing. Now, if we go down and we look at prophase, this is phase 2, which is the real phase. Okay, in prophase, if you look at the cell, all right, all the other organelles tend to sort of disappear. We're only going to draw what's happening in the nucleus. And it's not because everything else just goes away and the nucleus takes over the cell. We do it so that you can see on the diagram what it is you're supposed to see. Okay, so we focus now on the nucleus. So you've got your cell and you've got the nucleus. So in the nucleus, there's your nuclear membrane. We're going to have the centrosome, which looks like a little T, 2 little logs that make up a T, all right? Okay, the 2 little parts tend to separate out so that we end up with one of them here and the other one over there. So they separate out. And they start to move to opposite poles. Okay, so that's something you can see. Then you also are going to see the chromosomes, and the chromosomes now start to separate out. But they're still scattered all over the cell. So you see there's no real order here. If we looked at something, a cell undergoing mitosis in our bodies, we have 23 pairs of chromosomes, okay, always in pairs. If they aren't in pairs, it is a problem. So like, for example, Down syndrome, the child will have 47 chromosomes, which means they've got 46, which is 23 pairs, and an extra one, which is the extra 21st chromosome. They'll have an extra one. Instead of having 2, they'll have 3. And that's a mess up. We don't want that. All organisms on this planet have chromosomes that are in pairs. And if they are in pairs, they're always going to have, they go in 2, 4, 6, 8, 10, so multiples of 2. Very important for you to remember, because if you get asked to draw mitosis and you draw any one of the phases and you draw the chromosomes in an unequal number, it's wrong. Why? It can never be in an unequal number, right? Always in equal numbers. Now, during prophase, our chromosomes separate out so we can actually see them. But can you see here, the chromosomes are scattered randomly. Okay, so they're all over the show. Then, metaphase is wonderful because meta means middle. So it's always easy to pick out the metaphase. And if we look at metaphase, your chromosomes are all going to be lined up at the equator. Okay, I'm only going to draw, mind you, I better draw all 4. 2, 3, 4. Okay, and they lined up in the middle. The centrioles will be at the end. You're going to have your chromosomal fibers. Okay, like that. So your chromosomes are lined up in the middle. So you can identify it, because if it looks like nothing's happened, it's interphase. Preparation, chemical changes. If it's prophase, the chromosomes are there, they've lost water, they've shortened out, you can identify them, but they are randomly dispersed in pairs. Okay, metaphase, they're all lined up in the middle. All right, now the duplication is there, it's happened. Anaphase is easy, because if we look at it, there's the nucleus, centriole, we've got our spindle fibers, okay, and we're going to have our chromosomes pulling that way and our chromosomes pulling this way, okay. So they orderly. Can you see here? We have a little gap here in the middle. So there's a little gap. Why? Because the chromosomes are all pulling towards the end. If we look at late anaphase, you're going to have your chromosomes all sitting here like this. So they've pulled towards the ends. All right? But there's order. And then lastly, if we look at telophase, that's our last phase. We start with an invagination, or in a plant cell, you'll start with cell plates forming, okay, to separate the two cells. So here you'll have the centriole, you'll have your little chromosomes all gathered around the top here. And the biggest thing with telophase is this. You have that invagination, which now starts to divide the cytoplasm. And that is called cytokinesis. And what do we end up with? What is the result? The result is 2 identical daughter cells that are diploid. In other words, they are going to be 2N. They have a full complement of chromosomes. So if they started off with 23 pairs, there will be 23 pairs there. If they started off with 10 pairs, there will be 10 pairs in each of those cells. They are diploid. So they have a full complement, and we have identical daughter cells. Cytokinesis is a term they always ask. That's the splitting of the nucleus. And during metaphase, once this split starts and these chromosomes start to now pull towards the opposite poles, as that split starts there, we say that that is karyokinesis. And karyokinesis means the split of the nucleus. Okay, so as those chromosomes pull, it's the split. We're going to have a break?
Let's take a break. Okay, guys, as soon as we get back, we're going to be going through more of your questions that you have posted on the wall. The page has gone crazy. Grade 11s, you have showed up. Yeah, awesome, awesome. So, guys, don't leave those seats. Stay tuned to learn more and learn extra. See you now. Hi guys, and welcome back to Learn Extra Live. It's Life Science Grade 11, so I hope you're all tuning in. I know there's a whole bunch of you on the Facebook page and on Twitter screaming out questions for Kathy. There are a lot of questions on the kidney and the nephron system. I know I've got Faith, I've got Devishan, Neo, Njabulo, Mohammed. We are about to answer all of your questions. So guys, chat to us on facebook.com/learnextra, also at learnextra on Twitter. Kathy, I think we should get straight on to it.
Yeah. One of the questions was the difference in diastole and systole in the heart. All you've got to remember is when you die, you are completely relaxed. Okay? So diastole or diastole, doesn't matter, I don't care. Just write it, say it so you can spell it, okay? Diastole means that the heart muscle is relaxed, okay? And systole or systole is when it is contracting or working. Okay. So, use your common sense. The heart is made up of 4 chambers, all right? You've got the 2 top chambers and the 2 bottom chambers. The 2 top chambers are atria, and the 2 bottom chambers are ventricles. So, atrium, ventricle, ventricle. So, when the blood comes into the heart, it's coming in on the right, into the right atrium, okay? But it's also coming in from the lungs into the left atrium. So the atrium have to be relaxed. So we have atrial, diastole. Okay? Why? That's when they are filling up with blood. All right? Deoxygenated blood on the right, oxygenated blood on the left into the atrium. What's happening to the ventricles? The ventricles are just chilling. Okay? They're just relaxed. So, atria relax. What's the next step? The next step is going to be when the atria push the blood from the atria into the ventricles. Now, people, it's not a long distance. I mean, like, it's right there. So it's not like you need a heck of a lot of contracting. So next step is going to be atrial systole. But at the same time, while atrial systole is happening, we need ventricular diastole. Now think about it. It's going from the atria into the ventricle. So watch my hand. Atria or atria ventricle. Okay? The atria is cool, relaxed, so is the ventricle. The atria fills up. As it fills up, it's quite relaxed. There's a little bit of pressure that now occurs on the walls, but nothing major. Then it's going to contract. When it contracts, it squeezes the blood from here into the ventricle. Chips. Ventricle has to be chilled. It's got to be completely relaxed. So in diastole, when the atria contracts, it is in systole. Okay, so now that's step 2. Now the blood has gone from the atrium, shoops, into the ventricle. Now we have the biggest issue. And this is when step 3 takes place and we end up with the ventricular systole. Why? Because the ventricles are now going to pump the blood from the right side, the right ventricle, it's going to go to the lungs to get oxygenated. So that's not a very big distance. But the blood in the left ventricle, is going to have to go out through the aorta to every single part of the body via systemic circulation. So that is one heck of an amount of pressure. So what happens? The ventricle or ventricles are now going to contract, and they're going to contract like you have no idea. They go, whoosh, and as they contract, we now have ventricular systole to push the blood wherever it's got to go, either to the lungs or to all the parts of the body. And that is why the left side of the heart muscle is thicker than the right, because the left has to pump a heck of a lot of blood to a lot of places. Okay, but at the same time, why have you moved? At the same time as all of the ventricular systoles taking place, we are going to have atrial diastole. Why? Because when that blood pushes from the ventricle up into the atrium, what happens? The valves, those semilunar valves, okay, I'm sorry, the tricuspid and the bicuspid valve between the atria and the ventricle, they get clapped closed to go claw, and that is the loudest part of your heartbeat. You know, when you listen to your heartbeat, it goes doof doof. That's your loudest part. That's when the pressure from the ventricles closes the tri and bicuspid valves between the atria and the ventricle. Oops, closed. Now the blood goes spewing out, and it goes up out of the heart to the lungs, okay, and out the aorta to the rest of the body. And then what do we have? Now the ventricle's contracted, the atria is relaxed, and then the ventricle relaxes. And we have ventricular diastole. Oh man, diastole and atrial diastole. And when we have that ventricular diastole, there's nothing forcing that blood out anymore. So what happens? It slows down and it stops as the ventricle relaxes. And when it relaxes, there's no more pressure. And the blood now starts to flow back in via the pulmonary artery, okay? And it starts to push back from the aorta, that way, all right? And what happens? It says, hold on a minute, there's no pressure. And as soon as there's no pressure, the pulmonary artery and the aorta both have a semilunar valve. And those valves sit like this. So while the ventricles are pushing the blood out, cool. But when that pressure starts to get less and less, the ventricles close as the blood starts to flow backwards. And that's the soft sound. That's the dub sound that you hear. So the lub sound is when the tricuspid valves close, all right, between the tri and bicuspid valves, between the atria and the ventricles. And the soft sound, the dub sound, is when the semilunars in the pulmonary artery and in the aorta close because of a lack of pressure. So that's your lub-dub sound. That's your difference between diastole and systole, and I've just done the cardiac cycle with you, and all of this takes 8 seconds. So that is why we end up on average with about 72 beats per minute. Okay, now let's do the kidneys. We got 10 minutes, huh?
Mm-hmm.
Alrighty, I think I need another color. I'm getting a bit tired of this yellow. Let's go with pink. Okay, we have the kidneys. The kidneys form part of the urinary system, okay? And the whole process that they fulfill, or the function that they fulfill, is excretion. We excrete from our lungs, okay? We excrete from our kidneys. We excrete from our liver into the blood, but that the substances that are excreted by the liver, for example urea, is in an insoluble state, so it can't hurt us. And it's the kidney's job to filter it out. So the liver sort of dumps inside. It's like us working in the kitchen, and you put rubbish in the little kitchen bin, okay? And then that rubbish goes to the big bin, which is outside, which is your proper big green bin that we put into the road to be collected. So, and that's the kidney's job. So your kidneys, your lungs, and your liver are excretory organs. All right, people, they get stuff out of the body that will harm us. What's the difference in a secretion and an excretion? A secretion is something that's good. It's got value to the body. It helps the body to work properly. Hormones are endocrine secretions, and all your enzymes, saliva, tears, serum, anything that is secreted, okay, and that is good, that comes from an exocrine gland because it is a duct. Endocrine glands, only hormones into the blood because they are ductless. Sure, I'm going through a whole bunch of things. I hope you're keeping, because that's like 3 sections we've just done there, quick. Okay.
I know, I'm actually, as you're going along, Kathy, I'm going, I have to ask Kathy about this question, but as you're doing it, Kathy, you are actually answering a lot of the learners' questions.
Cool. Okay, people, but keep asking. All right, so we've now got our excretory system, and we're getting excretion is something that's bad. It's not good for you. We need to get rid of it, okay? We also say the skin is an excretory organ because we sweat, and the sweat, although it is good, so therefore we would say it's a secretion because it cools us down and controls our body temperature, but we are using waste substances to cool us down. Okay, and that it's because it's waste coming out that we say the skin is also a form of an excretory organ. Okay, so now if we look at excretion, we have the urinary system. 2 kidneys, left and right, okay? Then there's a tube that comes from those 2 kidneys. So kidney, all right? We've got this tube that comes out of there. This lumo actually looks quite nice. And that goes into the bladder. Now remember, a bladder is nothing more than a bag. It's just a bag. It's a holding bag. So that should be a kidney. These 2 are ureters. And what comes out here to the outside is the urethra. Now, ureters and urethra is something that learners tend to confuse and mess up. And here's a way to remember it. When the urine goes from the kidneys down the ureters into the bladder, it's soft. It goes in little drops. So it goes down the ureters. It's a nice soft, just imagine all these little drops of urine coming into the bladder, okay? But when we want to get that urine out, what do? We thrust it out, okay? So the urethra thrusts the urine out. If you have wanted to go to the toilet for 2 or 3 hours and you finally get to go to the loo, I promise you now it thrusts out. So remember, ureters, there are 2 of them, and it's drip, 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 drip. It's soft. Ureters. Urethra thrusts it out. You only got 1 tube, so it gets thrust out. All right? The bladder, nothing happens in the bladder other than to store the urine. We have a little valve here called the urinary valve, and that valve is what we learn to control by the time, hopefully, you were 2 years old. If it was later, shame. If it was earlier, so what? Okay, but as long as you can control it now, then that's cool. All right, so there's the urinary system. Now, if we take the kidney, come. I think I've reached the end of this page. If we take the kidney and we look at the macroscopic structure, what will we see? In other words, if you take a kitchen knife and a sheep's kidney and you cut it in half with a really sharp kitchen knife, what are you going to see? You're going to see a structure that comes out like that. You're going to see two blood vessels. The one on the top is the artery, and the one on the bottom is the vein. How do you remember that? A comes before V in the alphabet. And it's to do with the kidney, so they will both be renal. Anything to do with the kidney is renal. Renal artery, renal vein. Renal artery will bring blood to the kidney. Renal vein takes deoxygenated blood away, but clean blood away from the kidney. So the artery is going to have wastes, oxygen, nutrients, that's coming in. From the vein, we are going to have cleaner blood, so there's going to be less wastes, there's going to be slightly less nutrients, and there's going to be carbon dioxide. Right. Why slightly less nutrients? Because the kidney is like a chef. The chef eats while he's tasting all his food that he's preparing. So by the time he's prepared his meal for his restaurant, he's not hungry anymore because he's been eating little bits all the way through. And that's what the kidney does. The kidney literally takes its nutrients from the blood it works with. All right, so cleaner blood from the vein, wastes in the artery. This structure here goes to the ureter. All right, now if I look at this kidney and I cut it open, I'm going to see two very distinct areas. This area here that I'm putting little dots on is going to have a sort of a lumpy, granular texture. And if you look at it, I promise you now you can actually see, even if you've cut it with a not so very sharp knife, you will see that it's lumpy. It's lumpy and it's also very dark. So it's got a dark, it's a dark color, and the dark color is because all the blood's sitting here, and that dark colored area is called the cortex. Now the cortex, people, in any organ is always on the outside. The cortex is on the outside, the medulla is in the middle, medulla is on the inside. Okay, so you've got this granular cortex that looks dark. It's a dark, actually a purple red. So this dark maroon, dark purple red, and it's lumpy. And then you've got this area here, which is beautifully soft. And that's what you'll see. Okay? Now, what are these areas here? Each of these little triangular structures is called a pyramid. Okay? And this area here is in the middle. And that middle region, this middle region here, we call the medulla. And the medulla is made up of all the tubules. And because it's made up, tubules. And because it's made up of all those tubules, it has a very light color. Okay, and you can see it. It's like a pretty, it's actually a very pretty pink. So it's a light pink because of all these tubes, and those tubes contain the urine, which is full of all the bad stuff that we must not get out of our body. Okay, I think we're due for a little breaky break.
Yeah, let's take a little break. I'm wondering if I can ask you a bit of a risque question. Don't worry, producer. What about that fad that when people drink their own wee? Is wee bad for you?
You have no idea how that caused immense issues with so many learners. That program was broadcast just before matric finals. And
saying wee is good for you.
And they're saying urine's good for you. Geez, drink your own wee. And you had this man and this woman in the morning, and before they were about to brush their teeth, there was their nice warm cup of first morning wee. And they,
delicious.
I'm going to vomit. But that's what they did, because what you're doing is you're poisoning your body. And the body works and a negative feedback. So when you really damage it in one place, it will compensate in the other place. And that literally is what happened. Is you poison your body to such an extent it thinks, oh my goodness, and it then works really hard to try and correct that balance. And in doing that, you end up being okay. But it's not a good thing. That's why people who are in the desert and they're walking for miles and miles and they have no water, if they drink their wee, they actually eventually poison their bodies. Okay, so it's not a good thing.
Yeah, so
don't
do
drink
it.
your urine. And by the way, wash your hands after you've finished as well.
Good tips, all of these. Okay, guys, we're going to take a quick break. Make sure you get some water, some tea, jump up and down, get a little bit of energy going within you. Okay, see you now after the break. Hi, grade 11s. Welcome back. This is Life Science. Just in case you're just tuning in, you're a bit late, but don't worry. Kathy is going over any question or anything that you have been struggling with this year. So it's all about revision. Talking about revision, guys, we have winter school coming up. And for the grade 11s, it's going to be pretty awesome. Let me just have a look here, and that's going to be over the week of the 9th of July to the 12th of July. And what this is this is a winter school guide, and what it's got awesome questions and things you can work through while we are in the live shows.
Can I see? Because this is the first I'm seeing of this. It's so nice.
Prachtig,
these
nee.
are all my questions from the recorded sessions.
Yes. Kathy.
Okay. Do you know who's going to be doing them?
No, we don't know who's doing them yet.
Oh, okay. Guys, you know what? I have no idea who's going to be doing them because I'm certainly not going to. But I would like you to send all your questions from your June, July exams. Send them through. Okay. And best, I think, if you send them through to Indy when the session starts, I'm sure whoever's doing it will get to them and explain to you where you got all your marks that you did, because we've done such a good job during the term.
Yeah.
Okay. Huh?
Okay.
I think it's so nice, eh?
Kathy.
All right. Okay,
Let's get to it, guys.
Right, now, somebody asked, and I'm going to do this quick, asked Indy about if you do exercise, do you produce less or more urine? Think. When you do exercise, your body heats up. How does our body keep cool? It sweats. So, on a hot day, you are going to make more sweat. All right, so if you're making more sweat, what do you need to do? You need to keep more water in your body. So therefore, we are going to have more concentrated urine. All right, it's going to be very concentrated. Whereas if it's cold, you don't need to use water to make sweat, because guess what? You don't need to sweat to cool your body down. You need to warm it up. So what happens? You will make more wheeze, and your wheeze will be very dilute. Same difference as in the morning when you wake up, your urine is very yellow. And if it isn't, well, then you got up 5-6 times during the night, or you went to bed very late and you drank lots of water before you went to bed. Okay, but just on a normal process, if you went to bed and you slept for 7-8 hours, your body's busy cleaning itself. So, and you're not taking in any liquid while you're sleeping for 7 or 8 hours. So your urine will be more concentrated. Okay, the body will have time to clean itself out and reabsorb the water. And that is also, you've got now 6, 7, 8 hours worth of urine sitting in your bladder. That's why that first wee of the morning is always darker. Okay, whereas your last wee of the day, if you can even distinguish color, you haven't had enough liquid. Okay, so that last wee of the day, you know, I've now had enough water today and my body's completely hydrated, when there's very little color difference, and there should be absolutely no smell ever. If you can smell your urine when you're making a wee, you have some kind of infection. You need to get to a doctor and have it checked out. Okay, all right, so here we go. We've got the cortex and the medulla. So if we look at the kidney again, there's the cortex, and it's dark, and it's granular, and then we've got the medulla region here. Okay, the medulla is made up of all these beautiful little tubes that then go out via the pelvis. You've got to learn those labels, people. I'm going to write you, learn your labels. Okay, now Why do we have this difference in color? The medulla is made up of the tubules, and the cortex is made up of a very, very special part. Now, what we're going to have here, and I'm going to do it like this, okay, is a little thing like that, and it goes wiggle woggle, and it drops into the medulla, and up again, and wiggle woggle, and back into the medulla. Okay, that little thing that I've drawn here is called a nephron. And each kidney has thousands and thousands of nephrons. Why? Because the nephron is called the filtering unit of the kidney. It's the nephron that does the job. It's the nephron that does the work. How and why? Now we're going to do that. Okay, so we say, right, if I look at the nephron, you've got this little bowl, and it goes wiggle woggle, drops down, comes up, wiggle woggle, and down. So, and then it goes to here, then it goes smaller. Now remember, whenever a tube goes from big to small, we're going to have an increase in pressure. Always, okay? Now the same is if it goes opposite. If it goes from small to big, well, then we have a decrease in pressure. Okay, so what we'll have here is another tube and another one from there and another one from there. And this section here is called the collecting tubules. And each little tubule is going to, this one here will come from another nephron and another nephron. So each of these collecting tubules come from different nephrons, and they all collect into one tubule, which will then form part of the pyramid. Okay, now, something else that's very important is this part of the nephron is in the cortex. And this part is in the medulla. And that is very, very relevant. People, you must know that. This part is in the cortex, this part is in the medulla. Now, what do we have? We've got, first of all, this little section here. And this is where the first process occurs. Then we've got this area here, which is where the second process occurs. The third process, the fourth process, and then the fifth process. Okay, so we're going to go through them. And this, if I was asking you questions, I would make sure that you knew this part here the best. Okay, because we haven't got enough time to go through it all. So this little thing is called the Bowmans capsule. Bowmans capsule is like a little bowl, and in that little bowl, it's got a whole bunch of capillaries with a larger arteriole coming in called the afferent arteriole. It arrives, and this is our last stint. We now go to matric. Say, oh, good Lord. Okay, so you got the afferent, starts with an A, it arrives, it's slightly larger, the efferent leaves with an E, okay, for exit. The afferent is larger than the efferent, and that creates pressure. And that pressure is for a process called pressure filtration. And that you must know. So the afferent is larger than the efferent arteriole, and that creates pressure for pressure filtration. And then in that same little structure, you've got podocytes. And those podocytes are for ultrafiltration. And what do? They make sure that your red blood cells and the fats and proteins are too large to pass through into the Bowmans capsule, so the body doesn't have to wait to try and get them back into the blood. So they stay behind and carry on via the efferent arteriole. And that's why we say ultrafiltration. So it's water and all the little things that go into the kidney, so we can reabsorb the good things and the bad things go out.
Okay, phew. Okay, guys, grade 11s, we have come to the end of our show. And Paul, I know you said I wish the grade 11 show was on for an extra hour. I completely agree, if only. Okay, guys, this winter school grade 11 textbook, or should I say guide, has been given to Princess Ndix. I'm not quite sure how to say your surname, but I will send you a post on Facebook informing you of your win. Thank you so much, grade 11s and grade 12s. You better stay tuned for to you guys next. See you soon, grade 10s. Learn more, learn extra. Bye.